Hello students, this is the sixth video of chapter Strategies for Enhancement in Food Production, and topic of today is plant breeding for disease resistance, in which we will study method of breeding plants for disease resistance. So let's start. So what do you mean by disease resistance? Disease resistance means the ability of the breeder to prevent pathogens from causing disease in the plant. So it is the ability of the breeder to prevent pathogens from causing diseases in the plant. What are the advantages if we make a plant disease resistant? Number one, it would enhance the food production by reducing losses due to disease. So food production would increase because there would be reduced losses due to disease. And secondly, there would be reduction on dependency on use of fungicides and bactericides. So we would be less dependent on fungicides and bactericides. Now let us look at some plant diseases which are caused by fungus, bacteria and viruses given in NCERT. The three fungal diseases are red rot of sugarcane, brown rust of wheat and late blight of potato. These are the three fungal diseases. Black rot of crucifer is the bacterial disease, and tobacco mosaic and turnip mosaic are the virus diseases. Now let us look at methods of breeding plants for disease resistance. There are four methods of breeding plant for disease resistance. First is the conventional method of breeding. Second is mutational breeding. Third is genetic engineering, and fourth is selection of soma clonal variant. These two methods we will discuss in this chapter. Rest we will discuss in biotechnology. So let's start conventional method of breeding. So in conventional method of breeding, we do the following steps. Number one, we collect the germplasm. Germplasm means diverse allele of a gene for a crop. Secondly, we select the parents from these germplasm. Then we cross between these parents. Then we select the progeny which is obtained as a result of crossing between the parent, and at last we test the progeny and then release it in the market. Now let us study the second technique of plant breeding, that is mutational breeding. Mutational breeding is the technique that helps to create new trait originally not found in any of the available variety. So in mutational breeding, we create new trait or characters that is originally not found or present in any of the variety. Mutational breeding can be induced by chemicals or radiations like gamma rays. By mutational breeding, we have made mung bean resistant to yellow mosaic virus and powdery mildew. Now let us talk something about Parbhani Kranti. The wild species of plant had one advantage and one disadvantage. The advantage of wild species of plant were that they were disease resistant, but the disadvantage was that they had low yield. So we can transfer this disease resistant gene from the wild species into a high yielding variety, so that it can become both high yielding as well as disease resistant. The example of this is Parbhani Kranti. Now what we did is we transferred the resistance of yellow mosaic virus from a wild variety of bhindi into a new variety, and hence we got the variety known as Parbhani Kranti. So if you have gained something from this video, please subscribe now to my channel Ramji Chopra, comment, like and share.